So teaching is really a wonderful experience to learn. So without taking much time, so I will start my session on DNA evidence in the era of modern bioforensics. See, DNA forensics is not a new term, right? It is a very old term, right? Since I think 1985, people are knowing about DNA forensics and the use of DNA evidence in forensic investigation or any kind of criminal investigation. Okay, so let us start my presentation. So I will share my presentation. Just a minute. I hope now it is visible to all of you. Vikas, is it visible?
Yes, sir. Yes, sir. Sir, just 1 minute, sir. Sorry for the interruption.
Yeah,
Dear
no problem.
participants, please pin to the presenting ID of Dr. Bhargav Patel so there will be no disturbance between the session. Thank you. Sir, you can carry on, sir.
Okay, so dear audience, I know you have spared your valuable time to listen me, right? So it is my responsibility to satisfy you with whatever the knowledge I am having. I will try my level best to introduce DNA evidence to all of you that you can basically understand, because see, right now we are having a mixed crowd. So I will be discussing not in much more detail, but yes, of course, I will be discussing about the basic fundamental part of the DNA evidence, and then I will discuss about tools and techniques. What are the modern techniques which are now available and still it is not being that much exploited? And finally, what are the challenges with the DNA evidence that I'm going to discuss? So these are my, just a minute. So these are the contents that I'm going to cover. Right. So this is introduction. So I will introduce first with this DNA and what is forensics and what are the sources of DNA. So it is very much important to know what are the sources of DNA in order to understand how DNA evidence works on the crime scene. Then DNA evidence, what are the myth and truth? See, certain myths are there which are always, the DNA is always exaggerated, right? So that I'm going to discuss today. What are the different technologies which are nowadays utilized by the forensic experts to use DNA evidence for criminal investigation? What are the modern tools which are waiting to be used till today that I will discuss? And finally, I will conclude my session with the challenges with DNA evidence. So let us start with the introduction. So whenever any technique is discussed, so definitely the founder must be remembered. So I am thankful to Sir Alex Jeffries, so who invented this technique in 1985 when he was searching about some disease-related genetics in human. So he was approached by one of his friend, police officer, who was appointed as an immigration officer at the courts. So at that time, one case came about the Ghana immigration case. I think many of you are knowing about this case in 1985, and that case became the foundation stone for the genetic analysis for forensic investigation, right? So the Alex Jeffrey used a technique was known as restriction fragment length polymorphism. This technique is depending on the parts of chromosome that are continuously repeating for many times, right? And he exploited these particular variations among the human for the foundation of this genetic testings. So he realized that some part of the DNA are having some repeats, and these repeats are variable from one person to another person. So you can see here there are 3 repeats, and here there are more than 3 repeats. So on one chromosome, you are having 3 repeats. On another copy of that chromosome, you are having more than 1 repeats. So this condition is known as heterozygous condition. And here you can see you are having equal amount of repeats on both the chromosomes. I think everybody must be knowing that we are having 23 pairs of chromosomes. So every chromosome is in a pair, having, you can say, a counterpart which is completely identical. Like you can say having identity, but you can say variability is also there. So this kind of variability accounts for heterozygosity, and this kind of similarity accounts for homozygosity. So when you are taking the DNA from both the person, and when you are designing a probe which can bind such kind of DNA part, then you can do this kind of analysis. So what do? You chop the DNA in small pieces by using restriction endonuclease enzymes. So at that time, this enzymology was very known, and cloning techniques were also very known. So enzymes were used to cut the DNA. Nowadays, the cutting of the DNA is done by some other modern tools also. After the cutting, when you are doing the electrophoresis, then all the smallest fragment to the highest, you can say the largest fragment will be arranged in this fashion on electrophoretic gel. And from gel, you are transferring this particular bands to the membrane, nitrocycline or nitrogen, whatever the membrane you would like to have. And then these membranes are flooded with the probes, which are specific for that particular sequence. And then these particularly, Alex Jeffrey used radioactively labeled probe. At that time, I know that fluorescence probes were not available. So only radioactively labeled probes were used. And then after that, if you are creating an image on X-ray film, this image will look like this. And this image was looking like, you can say, barcode of any product. I think many must have visited a supermarket in which barcodes are there, right? So similar, it is looking like similarly. You can see here, the person A and person B are different in the bending pattern. So this is this is because of the probes which are binding to the different cut pieces of the DNA. So this variability stands for the uniqueness, or you can say the uniqueness of the person. This person is unique and this person is unique. So both the persons are not related. You can see here. See, some bands are similar to each other, but see, these two bands are not matching with this another person. So what does it mean? It means both the persons are different persons. But here you can say this particular was multi-locus fingerprint. It means one probe was binding to at many sites on the probe. So on the particular gene or marker that Alex Jaffrey wanted to bind. But later on, he found out a solution to make it much more clearer than single locus probe. It means it will bind only on that particular gene and it will never bind any other site. So more specific probes were designed, and finally you can get only two bands if it is a heterozygous, and if one band only if it is homozygous. So this clear the doubt, or you can say this is very easy to interpret. This was the fundamental technique by which Alex Jeffrey used to solve the cases. But nowadays in the modern forensics, this kind of analysis is basically done. You can see here, here are three persons profile I have given here for one chromosome. You can see here chromosome number 1 are in a duplicate. This is the one piece of chromosome and another piece of chromosome. It means both are in a pair. If you will see that both are having almost similar genes, but it may vary for different parts of different genes also, or it can contain different alleles. This is called the heterozygosity. You can see here this person is having AGAT. So this AGAT is nothing but a short tandem repeats. So these are called from starting from 2 base pair to 7 base pair. These particular small fragment of DNA are continuously repeating for a longer period. It may be 4 times, 5 times, 6 times. You can see here. Here, this person is having 7 repeats on one chromosome and 6 repeats on another chromosome. So here, this woman is having 4 repeats and 19 repeats on another chromosome. Here, it is 12 repeats and 14 repeats on another chromosome. So indirectly, you can say that this person was having this chromosome from his mother. If then his mother was also having 7 repeats, right, on this particular chromosome. And if this chromosome is from father, then 6 repeats from his mother, right? And this is how he becomes heterozygous for this particular position, right? So these kind of markers are analyzed nowadays in forensic science, and based on these repeats, the sizes of the product, the PCR product, is analyzed in electrophoresis, and finally the STI profile is generated. I will discuss STI profile later on, but This is the fundamental. So these kind of markers are identified, which are universally available in the human, but are variable. It means it is having more than one allele. Some markers are having 30 alleles, some markers are having 40 alleles, some markers are having 80 alleles also. So what does it mean? 80 alleles, if one marker is having 80 alleles, what does it mean? It means you can identify 80 homozygous persons and more than 80, right? Or 80 minus 1 divided by 2, 80 into 80 minus 1 divided by 2 heterozygous persons from that, using that marker, right? So it is just like the person's identification based on their appearance. For example, you are wearing a hat, right? So hat may be of different color. For example, 100 color shades are available. Then if the person is telling that I have seen that person having a hat, but which hat? Blue hat, white hat, green hat, which hat? Right. So what does it mean? It means you are having 100 variability. It means 100%. Now you add another marker. The person was wearing hat also, and the person was having goggles also. Right. Now you are having two markers. The person who was having hat and goggles, but pink hat and which goggles? Blue hat and goggles, green hat and goggles. So these are the combinations of the marker. So this is how you increase the marker, the uniqueness of the persons will be increased, and finally you can identify the same person which was involved in any criminal activity. So this is the basic fundamental of the short tandem repeats. So this is the 13 Cody score loci. Basically in 1996, the US, United States of America, decided these 13 markers to be the minimum number of markers to be analyzed for any kind of DNA analysis. Basically, the DNA forensics started in United Kingdom because Alex Jeffrey was from UK, and they have started very early. But later on, many companies started this particular business, and finally, US was the biggest. You can say the biggest country who started the countrywide. You can say analysis of the populations for this kind of marker analysis. They have found out number of other markers also, and finally decided. So nowadays, apart from this 13 Cody score loci, we are having almost total, if you combine all the markers of all the kits from different manufacturers, we are having 35 to 40 markers which are available to identify a human, right? So you can see these are the position. It is on chromosome number 2, chromosome number 3, chromosome number 5, chromosome number. So you can see on chromosome number 5 you are having two markers, right? And many chromosomes are there which are having two markers, or sometimes three markers also, right? But most of the chromosomes are covered for different markers. Now, about how the DNA forensics, or you can say the STI profiling works. But now, what is the main important thing is the sources of DNA. See, what are the sources of DNA? So, if in biological language, if I'll tell you this, actually the cell is the source of DNA, right? A human cell, tissue cell, right? So, because our whole body is made up of cells, except RBC, red blood corpuscles. Except this RBC, all our cells are having nucleus, and it contains nuclear DNA. It contains mitochondrial DNA as well. So we are having two types of DNA, mitochondrial DNA and nuclear DNA. So both the DNA are having forensic importance. Mitochondrial DNA is analyzed in a separate way, and nuclear DNA is analyzed in a separate way. Whatever the STI profiling till now I have discussed is basically analyzed on nuclear DNA, or you can say autosomal DNA, right? So this DNA is found in a cell, right? And these cells are available on crime scene, right? On many objects, right? So this slide is representing the hands related stuffs which are having the possibility to contain DNA. You can see the armrest, automobile, baseball cap, binder twin, bottle cap, chocolate bar, cigarette lighter, cigarette paper, credit card, detachable box, magazine, pistol. Right. So many times, the ballistics instruments are also involved. Right. You can see the weapons are involved. So at that time, the object you are touching will always contain your DNA. Why? Because you are shedding your skin cells regularly, continuously. every day. Millions of cells you are shedding, right? And the new cells are being formed. And these cells are the source of DNA. And these cells can be trapped from on any objects. And that's why these objects are very much important. You can see the envelope, the self-adhesives. So when you are, licking that, the flap, and you are pasting it on the envelope, at that time your saliva will be trapped into that envelope, and that may be the source of DNA, right? So these kind of, gloves, handcuffs, ignition switch, keys, knife handle, magazine, paper, even the bullets, which is triggered, and which is used, that bullets are also having possibility to have the DNA, because when the magazines is filled, the bullets are handled by your barehanded. So that's why they make a rope, because when strangulation is done, rope is having a possibility to have DNA. Screwdriver handle, seat belt buckle. So these are the various, you can say, uh, parts or objects which can contain the hands related DNA sources, right? Moreover, I would like to tell here that many times these sources are always successful. I'm not telling like that, right? So, but there are possibility that you can find the DNA from these kind of objects. Mouth and nose related, right? You can see the airbag. When an accident occur, what happens if the car is sophisticated? Then airbag will be blown off. So at that time, your mouth, this head part will, will collide with that particular airbag, so that you will be survived. So during that particular accident, many a time the blood or saliva will stick to the airbag. So this is a very good source of DNA. So many times, in accident or insurance cases, it is very important to know whether the person was died because of the accident, or it was planted. Because many insurance companies to investigate this kind of, when there is a suspicions of murder, or suspicions of some kind of trapping, at that time this kind of analysis will help you, right? You can see the bite marks, which are also very well known for the DNA analysis. Cheesecake, chicken wing. See, these are the mouth related. When you are eating something, saliva will stick to that part, and when you are eating, non-vegetarian part, so you will leave that bone part. So that bone part will have definitely your DNA. So when you are having a crime scene which involves such kind of evidences, you can definitely take help of this kind of evidence. And cigarette butt is definitely regularly, envelope and lipsticks. So yeah, I remember one very interesting case. I think many of the students who are already listening this might have heard about this story. Actually, what happened in the US, when supermarket was small superstore was robbed by one robber, and the store was mainly managed by a lady. At that time, lady was there, and the robber robbed the store. He was learning person. Gloves were there, the mask was there, and completely was prepared. So none of the traces were there. Even the hair were completely tied, completely. So nothing. Even he broke the CCTV camera, so that even it cannot be also traced back. So finally, after robbing the cash counter, he ran away, but he came back after few seconds. And what happened? He kissed that lady on the cheek, and he ran away. And after 2-3 hours, when the FBI came, when it was reported, so they interrogated the lady. What happened? So finally, they came to know that he kissed that. So immediately they took the swabs and did the analysis, and by differential extraction and by eliminating the profile of the woman, they could find out the actual profile of the robber. And when they matched with the database, actually the robber was means previously convicted, so his DNA profile was there, and he was caught. So this kind of analysis will help, right? Then you can see the telephone receiver, straw, stamps. I think you must be knowing that after WTC event, there was a bioterrorism attack on US, that bacillus spores, right? That anthrax spores were sent in envelopes. So basically, they isolated DNA from the stamps of that particular envelope, and they could revive certain profiles, and based on that, they did some analysis, and they came to conclusion that these people are related to that particular region based on population genetics. So these kind of, toothbrush, toothpick. So these are the very much interesting uh, objects which can have your DNA. Now, general ball-related hair comb, you can see the buried remains, burnt remains, contact lens fragments, dandruff, embryonic cord embedded in paraffin. So these all are having a very potential use for DNA analysis. Tissue paper, wiping of underarms, of shirt, sweat, or even tissue paper you are using for your regular use. Paraffin embedded tissues, they are also very much important. Many in many cold cases, this kind of paraffin embedded tissues may help in the very analysis of DNA, because you can have this DNA from this particular very old DNA also you can find out. So before, this was about where you can find the DNA, what are the sources. But when you are having different types of body fluids on the crime scene, so which body fluid should be selected? It should be based on the amount of DNA. So this is the general idea, which is given by this table I have taken from a book, that Butler. So you can see here, liquid blood is having 20 microgram to 40 microgram of DNA per ml. Right, blood stain is having 250 nanogram to 500 nanogram. Liquid semen, you can see it is highest DNA concentration it is having, and that's why always it is preferred when you are having the rape or any sexual assault or this kind of analysis at this kind of scenario. At the time, liquid semen is the best source of DNA, right? Then plucked hair, shed hair, liquid saliva, oral swab, urine. Urine is having the least amount of DNA, and sometimes no DNA is found in urine because it is not having any kind of cells. If you are suffering for any kind of infections, then at that time your urine may contain some kind of pus cells or some kind of the cells which are liberated from the urogenital tract. So at that time you can have the DNA from that. Bone is having a very less amount of DNA, but this DNA is very interesting because it can be preserved for a longer period of time, and it can be well preserved because bone is a very you can say strong tissue, and it can preserve the DNA for a longer period of time. So whenever you are having the the body remains, only the skeleton or a part of bone, so at that time it can be analyzed for that. Now, so this is about the myth and truth about the DNA evidence. So I'm going to discuss about 3 to 4 myth and truth about the DNA evidence. So let us take the first. So there is a myth that every crime scene will have a DNA evidence. Everyone is believing, because thanks to CID and many crime scene investigation series and many crime scene investigation related movies and you can say the series, people are believing that every crime scene will have a DNA evidence. No, it's not true. It's true that DNA evidence can be found on crime scene, but not always, as DNA tends to degrade. Sometimes DNA is not available on the crime scene. For example, if it is a case of fire, if it is a case of blast, and sometimes the person is very much learned, and at the time he will never leave any kind of traces of biological fluids. So at that time he will not be able to get the DNA. So this is the one myth which is prevailing in the society, but it is not true. Second is DNA is the first evidence to be analyzed. So many people, everybody talks about that DNA. Why? So it's not like that, right? DNA is the secondary choice, basically. What is the truth? First preference is given to the fingerprint, being very simple to analyze and very easy to analyze and very cheap to analyze. So people will search for the fingerprint. Then they will search about the CCTV footages, call records, mobiles, and some other stuff. And when nothing is found, then they will search definitely for eyewitnesses. So these are the first evidences they will search for. And when nothing is found at that time, they will go for the DNA. Or sometimes, for example, if the case is rape, at that time to prove the rape, only DNA evidence is not. Obviously, DNA will prove the rape definitely. But when multiple persons are involved, gang rape is there, or the person is reporting the case after 10 days or after 15 days or after a month, then at the time it will be very difficult to get the DNA from this kind of crime scenes. So DNA is not the first evidence, it will be preferred at the last, because DNA analysis is not a very simple task, it is a very complex, and you can say depending, because it is a very sensitive, and that's why it is prone to degrade, and that's why sometimes you might not get the results properly. So this is the truth. Second or third myth: DNA is the absolute evidence. It means if the DNA evidence is collected, the crime would be solved definitely. It's not like that. DNA evidence is not absolute, rather it is very sensitive. And when the evidence is sensitive, the manipulation, the contamination and degradation chances are very much, and that's why DNA evidence should be analyzed very properly, and before that it should be collected and handled properly. So no 100% recovery of DNA is possible from every evidence collected. So it is not like that the way when you are collecting DNA evidence, then definitely it is going to generate the profile. No, it's not like that, right? Out of 100% cases, it may be possible that hardly nine, you are not getting 100% success. Some laboratories are having good success rate, but I don't think so. It is more than 80%, right? 80 or 85%, right? So many times, if the DNA is recovered, but STR profiling may fail, you are not getting any STR profile, and sometimes you'll get partial or mixed profile. So you can help. Even this partial and mixed profile, sometimes it is so difficult to analyze, so you cannot put it in the court, because it will not have a much weightage as an analysis, or as an you can say evidence. So the DNA analysis 100% depends on how the evidence is collected and handled at the crime scene. And that's why it is very much important how you are collecting the biological samples from the crime scene. You have to be very careful while collecting these evidences, and that's why the knowledge, the training, and the, you can say, the practice, or you can say the hands of the first responders are very much important in terms of DNA to be the potential evidence in the court. Right. Now I'm going to discuss about the technique. Okay, so what are the tools of the trades? So starting from the collection, preservation and transport of the DNA evidences, from then extraction of the DNA, amplification of the DNA, STR profiling of the DNA, and finally the interpretation. So all the steps requires multiple tools and technologies, and that I'm going to discuss now. So first of all, the swabs, right? So this is, you can say, the Brahmastra of DNA analysis, DNA profiling machine. This is the Brahmastra. Why? Because this is the particular evidence collection device which will gather all the cells from the crime scene. As much as the cells you are collecting, as much as DNA you are getting, and if the good amount of DNA is present, then a proper profile can be generated. So there are various different types of swabs are available, but very intelligently designed swabs are there. You can see here, this is from Copen, that you can say the 4 and 6 flocks, right? 4 and 6. So this is pronounced like forensics, right? Flock swabs, crime scene. So it is having, you can see this kind of, this is the difference between the cotton swab, traditionally cotton swab, and flock swab. You can see here the arrangement of the flocks in such a way that it is having the maximum binding capacity. So it will collect the maximum cells which is possible, and when you are putting in the solution, it will release also the maximum number of cells. So it will not contain the cells for permanently into that particular matrix. But here you can see here, when it is a cotton, you can say it is uneven, or you can say the matrix-like structure, and your cells will be trapped inside this matrix. And when you are doing this, definitely people are lysing the cells directly, directly with the lysis solution. But still, the recovery rate of the DNA from these flocks is always lower than this kind of flocks. So you can see here they are having some perforation mark here. So you can break the flock in directly tubes, and you can transport it very easily. And this is coming with the tube, which is having sometimes desiccators like this, and you need not to dry it also, right? You can see here different types of anatomic and ergonomic designs are available for collection, right? I think I'm not sure, but as far as my experience is concerned, I know that many of you might not have seen this kind of flocks even on crime scenes also, because in India, whatever the practice is there, they always think about this kind of swabs. Swabs means this, but this kind of swabs are also available. Very few people are knowing about this. So this kind of knowledge should be imparted to the first responders. Even they should be available. This kind of, you can see here, which is having a desiccant. So it can prevent the DNA even in a very, you can say, humid condition also. Because see, when the DNA evidence is collected, it should be dry. Why? Because bacterial contamination should not be there. And wherever the water is there, because none of the crime scene will be sterile. And after God, microbes are omnipresent, right? They are present everywhere, right? After God, they are the second populations which are available from Antarctica to every part of the world, right? So your every crime scene will have some kind of or some different types of bacteria, and they are going to harm your DNA because DNA is a rich source of food, phosphate, and carbon and all that. So that's why your DNA is going to be degraded by the bacteria. So that's why this particular kind of swabs are having many you can say antibacterial agents and antifungal agents which can prevent the growth of that. You can see these kind of special tubes are also coming that directly you need not to even worry about the contamination. So these kind of well designed and engineered products are available and which can be very useful for the collection. These are the the swabs mostly used in India. You can see these are the having tubes and the cotton, mostly the cotton. Nylon swabs are hardly used in criminal investigations. You can see this is I have taken from one research, very interesting recent paper. They have compared different companies, different swabs in the recovery of DNA. And it's a very interesting paper. And if you will see how many criteria they have compared. So by these table, you can understand the importance of this particular swab in the collection of this. Right? It is very important. Then another device is FTA card. Fitchco-Flinders Technology Agreement. That is the full form of FTA. Nowadays, even many scientists are are telling it fast transfer application also. But this is the full form of Flinders Technology Associates and Fitchco-Flinders Technology Agreement. This is a wonderful card. You can see which is having this kind of circles. Basically, this encircled area is impregnated by some chemicals. So when you apply your blood or any kind of body fluids on this particular circles, right, it will entrap the DNA by lysing the cells. So it is doing multiple tasks. It is collecting the DNA. Sorry, it is collecting the cells. It is lysing the cells. It is preserving the DNA, right? And you can recover the DNA from this card. So this card is having dual function: the storage of the DNA also and the extraction of the DNA also. So it's a wonderful card, and the I will tell you the up to 30 years. It is well established and it is proven up to 30 years the DNA can be stored on this card at room temperature if the card is preserved in a dry condition. Right? You can see here this is how the card is packaged. You can write whatever the information about that, or you can just stick the barcode, and finally the details are entered in the computer. So it is coming in different variations, single card, double card. This is classic card, right, which is having and it is having a pinkish color. Basically it is indicator card. So whenever you apply any solution body fluid on this, what will happen? It will turn white. So wherever the area which is applied with the body fluid or any blood or something, it becomes white. For example, if you are applying saliva, right, and what will happen? Blood is having red color, so it will be visible. But in terms of saliva, what will happen? Saliva will not be visible. So in order to get where exactly your saliva is there on this card, they have come up with a very unique technology, and this pink color will turn to the white color. So these kind of devices are available. So after this, the analysis of FTA card is very easy. You punch the card, you take 2 mm. So out of this particular circle, you require, this is actually 1 centimeter circle, and out of this 1 centimeter, you require only 2 mm, punch of this, and the 2 mm punch is more than enough for profiling of any DNA. So most of the time, this card is utilized for the reference samples, or you can say the samples are which are recovered from suspects or the victims, right? And many a time when you are having a very good, not degraded but good amount of sample available on the crime scene, so at that time these cards are utilized. Some other modern equipments are also available. You can see here the saliva DNA collection wire. So here you have to spit into this particular column. Your saliva will be go down and completely preserved in this particular tube. And after this, you just turn it around and it will lock the system. And you can preserve the saliva at room temperature without any loss of DNA for longer period of time. So it will enhance the you can say the utility for transportation at room temperature, right? Because in a country like India, for example, if crime happened at remote area, so at that time you can collect the sample from the victim or collect the sample from suspect directly. If you don't have any swab available, you can use this kind of device to collect the saliva. You can see this is the 3M trace evidence collection vacuum. So when you are having a very trace amount of the sample on the any inanimated very big table is there or wall is there which cannot be transported to the lab, at that time you can use this vacuum. It is having a very high vacuum system which is having, you can say, filter here. It is 0.2 micron filter, so it will trap all the debris here. So this will also trap the cells also, and this debris can be utilized for the DNA analysis. Even whatever the liquid you are even collected in this system can also be utilized for DNA extraction. So this kind of modern equipments are also available. Another is MVAC system. This is a wonderful system for DNA recovery from the clothes, right? You can see the clothes. So when you are having this, it is having a sterile buffer which can be pumped into this particular device first, and then the cloths will be wet with this buffer, and then it will resuck. It will create the vacuum, and the same buffer will be collected from this vacuum device, and this buffer is filtered through a filter system, and then whatever the filtrate you are getting, that filtrate and the buffer, it can be concentrated and finally can be analyzed for the DNA analysis. So this kind of very this even not only on clothes but even very you can use it on plastic surfaces, and even the paper surfaces also you can use it for the DNA collection. Everybody is knowing about the storage and transportation. So you know that chain of custody should be maintained with the proper sealing and barcoding if you are using the barcoding, or if you are using the sealing pattern. In India, we are using the you can say the seal method. So the same seal will be supplied with the evidence to check whether it is from the same police station or not. And mostly this particular bags, paper bags, mostly the paper bags for the proper you can say the moisture management. Silica gels are also utilized to minimize and pool packs. So instant pool packs are also utilized to transport the DNA. Now about the DNA extraction. Starting from the phenol chlorophomisomal alcohol, which is the gold standard for the DNA extraction method, and till today, even the modern forensic laboratories are also using the same method for number of samples. But for the high throughput, for the fast, and you can say the recovery of DNA in a very simple manner, people have adapted, or the labs have adapted some modern kits which are ready-made available from different companies. So most of the part of world, these three companies are, you can say, occupying the market. You can see the Thermo Fisher, Promega, and QIAGEN. So Thermo Fisher is having a range of DNA extraction kits, right? Starting from the DNA extraction. Here I have shown you the identifier direct PCR kit. So what do you mean by direct PCR kit? It means you need not to worry about DNA extraction. Collect the specimen, add your specimen into this kit, and directly go for the PCR. So it means no need to extract the DNA. But this kit works mostly for the reference samples and good quality samples. It doesn't work with you can say very degraded or sometimes having contamination or you can say the PCR inhibitors. Most of the time it works, but it can fail. So that's why it is preferred for some good samples. Here you can say the swab solution and case for direct. So Promega is also having such kind of solutions. So people are using that initially. I think 1 year back we went to the Rwanda for training that particular laboratory for 3 months. So at that time, see the whole laboratory using the Promega products, and they were using swab solution in caseworks for their routine analysis as well. So when we analyzed the total cost of that particular lab, and when we reanalyzed and when we gave the another reanalyzed workflow, the cost was reduced to 70%. Can you imagine 70%? So this is how you can say it depends on you when you should use this kind of modern kit and when you should use even a simpler kit which are available and which can give you the results also, right? And the third is Kaizen, which is silica gel based technology, and the columns are there and in the column, and it can be automated also, and it can be done manually also. So these are the three different kits people are using. In Thermo Fisher, you are having the pure link genomic DNA extraction kit. In Promega also, some other kits are also available for the DNA extraction. And in Kaizen also, you can say the DNEZ blood and tissue kit and other kits are also available for the DNA extraction. If you'll see the automation, because nowadays each and every laboratory is getting more than many cases a day, and that's why you have to analyze at least 30-40 samples a day. So at that time it will be very difficult to go manually. So that's why you have to depend on some automation solutions. So you can see here one machine which is available from Thermo Fisher is automate expression. So directly 13 samples can be analyzed on this after the lysis. See, and any automated machines up to the lysis step will be generally manual, right? It will not be automated. So yeah, some machines are there. For example, the QIAcube. This is the version of QIAGEN. But another version of the QIAGEN that is called Symphony, QIA Symphony, which is having even the lysis steps also automated. So it depends, right? But most of the machines are having lysis steps manually, and sometimes even the lysis step is automatic. You cannot, you can say, go for the automatic step because the sample is in a such a way that you have to do the manual intervention because sample processing will require. For example, your sample is on a very you can say complicated tool, so you have to collect it and you have to purify the if the blood is in soil, directly you cannot go for that. Right? Directly you cannot go for because you have to remove the soil particles, you have to centrifuge it, you have to wash it, you have to collect the cells, and finally, otherwise you will get contaminated because soil is having a very much contamination, a very much good concentration of humic acid, and which is very difficult to remove from the DNA extraction. So that's why you have to be very careful. So this kind of this technology is working on DNA beads. So you can say the paramagnetic beads, which magnetic bead based technology. Here it is Maxwell. This is also based on paramagnetic beads technology. And here in QIAcube, you are having two different types of solutions. Here, whatever the machine is shown here, which is based on the silica gel technology, which is having the complete workflow for DNA purification and elution directly. Okay, so this was about the DNA extraction. Then the PCR. So this is the genie of molecular biology. So it is wonderful, or you can say the fundamental process which revolutionized molecular biology like anything. Because of this particular technology, not only forensic science but each and every biological science field was affected, and because of that, the world has changed, and we could identify many, or we could understand about the amazing things about the biological science in a better way, right? And PCR. So here PCR is basically used to amplify those markers which I have shown you in that particular slide, chromosome number 1, 2, 3. So here we are not going to amplify the whole genome of the person, but we are going to amplify only few pieces of the DNA. It is just like that out of the whole book, we are Xeroxing only few pages, right? This is like that, right? So we are going to Xerox PCR is nothing but the Xerox machine for the DNA. So it is continuously multiplying, or you can say Xeroxing that part of DNA in a perfect way, the way a replication system is doing in our cell. Right. In replication, we are getting only one copy of DNA, but after the PCR, we are getting, millions of copies of that particular DNA. So this was after the DNA extraction, the DNA is getting amplified, or you can say the markers are amplified by the PCR. And after the PCR, you are having the gel electrophoresis system. See, this is nothing but DNA sequencer, or you can say DNA genetic analyzer. It is nothing but a sophisticated electrophoretic system, which is known as capillary electrophoresis system, which is having capillary. You can see here, small capillaries are there, very thin capillaries are there. This is basically 3530 system, which is having 8 capillary system. You can see here, it can be 8 to 24 capillary, even 96 capillary system is also there. So at a time, 96 samples can be analyzed in this system, and it can be. So the fundamental of this particular technology I have shown here. What happens basically, you can see here, your sample is here in this particular, the sample holder, or you can say 96 well plate. This is your samples, and this multicolor are nothing but your PCR product, which are having the dye, different colors dyes, right? And when it is entering to the capillary, and then you are applying the voltage, and after the applying of voltage, your DNA will be separated based on their sizes. So the smallest product will run faster, and the heaviest product will be at the later stage. And then here in the between, at the end here, you can see in machine, you can see here this part. Here it is your starting part, and then your product will run from here, and this here there is a detector. So the detector is nothing but argon laser. So when you are applying this laser here, it will fluoresce the dye which is joined with the PCR primer, and it will fluoresce. And whatever the fluorescence you are getting here will be collected by this prism and will be separated by the prism according to that frequency, or according to that wavelength. You can see here, and then it will be separated based on color and will be detected on the CCD panel. And according to the color, you are getting here the picks. You can say one by one according on the scale of time. So this is how you are separating the DNA molecule based on the color signal from chemical signal to the light signal. So this is how the electrophoretic system works. And finally, you are having this particular picture known as electropherogram. And based on this analysis, you can tell which product is present and which product is not present, and you can analyze this particular electropherogram, and you can identify which marker is present and which marker is not present. And finally, you can create a table in which chromosome number 1, which marker, chromosome number 2, which marker. These are the numbers you are, and you are having a DNA profile with you. So this is how the DNA analysis is done. Now, I would like to introduce one more instrument which is not widely used, but it is having a tremendous application in forensic science. It is called a laser capture microdetection. So when you are having a rape case, or when you are having a touch DNA related cases, at that time you can use this particular fundamental machine. This machine is basically invented for the oncology, cancer research, right? How it is working basically. So I am having a small video here. I don't know whether you will be able to listen the audio. Just a minute. Vikas, is it audible?
No, sir.
Yes.
No, sir. No, sir. It is not audible.
Yes or no? It is not audible.
No, sir.
Okay. Okay. So then I think it will not be audible. Anyway, so I'll skip this video, but I'll explain how it is working basically. So see, if you'll see here, this is a cap. Right. This cap is basically, I'll show you in video basically, whether it's not, maybe not audible. You can see here, the tube is directly inverted on this here, and the cap of the tube is having this kind of arrangement, and which is having a small membrane on this cap, and this cap is inverted. Basically, this is inverted microscope, and whatever the smear you have prepared on the slide, you can select whatever the area you want to capture. Here you can see here, right? And this is your area, and you have captured it, and the laser will cut that area only. You can see here in this picture, you can see here. So they have marked this area, the 1 2 3 4, and automatically the laser will cut this part, right, this part, and it will be collected on this particular cap. So even a single cell can be captured. So when you are having a gang rape case, so at that time you can have a post-coital swab, you can prepare a smear on the slide, and you can collect the individual spermatozoa, and you can analyze the individual spermatozoa for the DNA profile. Though it is a very tedious and you can say time-consuming technique, but in a gang rape case, or when multiple persons are involved and you are getting a multiple pick and multiple, you can say the mixed profile, at the time it is a wonderful technique to identify the individual persons. And because single cell genomics is a modern tool which is available, and you can amplify from a single cell also the whole genome, and then you can analyze. It may be time consuming, it may be cost effective, but when justice is concerned, you can use this particular technique. Still, other modern tools which are available and which are being used also, but not that popularly, is one, this called rapid hit technology. So this is you can say recently introduced by Thermo Fisher Scientific. I think before 2 years back, this technology was introduced. It is just the swab in, profile out technology. So what you are doing here is just you enter your swab. Basically, it is a cartridge-based technology. You are having a cartridge in which you will enter your swab you have collected from crime scene or whatever, the buccal swab. Mostly it is used for the reference samples, so buccal swab is there. And finally, after the buccal swab you enter, just click on this button. That's all. After 90 minutes, you will have a DNA profile on your computer. It means collect DNA extraction, PCR, and DNA separation. The PCR separation is done in this small chamber, capillary electrophoresis. It is based on microfluidic devices system, and it is doing in the same. So this machine can be used by even illiterate person who is having none of the idea about how DNA forensic works, but the policeman can collect the sample from the person and directly put that. So when you are having a suspect in front of you, you can directly collect the sample from the suspect, put it inside this on the police station itself, and send the profiling to the forensic lab for the comparison purpose. So this particular technology is adopted by US, it is adopted by Europe also, and many police stations are doing this work regularly, and because of this, conviction rate has increased like anything. It means because after the suspect is collected, the suspect is going on a parole, or if it is outside, then it may escape. They may escape, and it is very difficult to catch them once again. So when they are in the police station itself, you collect the sample and directly do the analysis. So within 90 minutes, it will be clear whether the suspect is the perpetrator or not immediately. So this is how you can do this. So this technology is very, I know it is costly. Per sample cost is not that much low, but it is available. Then next generation sequencing technology. See, NGS is available since many years, right? But in forensics, the NGS is still not that much preferred one because of its cost. Because nowadays, because this NGS technology will give you not only STR, but apart from that STR, it will give you the mitochondrial markers also. It will give you SNPs also, and other markers also. Because nowadays, whatever the panels which are coming for the next generation sequencing for forensic panels, which are having more than 300 markers. So what does it mean? It covers STRs, it covers other markers, it covers mitochondrial markers, it covers all the SNPs. So in a single run, you will get all these things, whether you want or not, right? So it will generate the complete profile of the person, So you can know the ancestry, you can know the individualization, you can know the phenotyping, you can know the management. This is phenotyping, ancestry, their health conditions, and STRs. Everything can be known in a single run. Right. So when some complicated cases are there, high profile cases are also there, at the time you can use this particular technology. So nowadays these 1, 2, and 3 profile, platforms are available. This pyrosequencing was the first platform to be introduced as an NGS. Nowadays it is considered to be absolute technology. But this sequencing by ligation and sequencing by synthesis, this solid platform, basically it is introduced by Applied Biosystem, and the Solexa technology, this is introduced by synthesis by the Illumina technology. So these are the both the recent advances. Apart from these, you can say the completely whole genome sequencing. Single genome sequencing is available. That is available by PAC Biosystem. So that is not that much useful for the forensic analysis, but just for your knowledge. Then from genotyping to forensic phenotyping. So forensic phenotyping is nowadays, the very important, you can say, uh, area where forensic scientists are working a very lot. You can see here nowadays, you can read this word high risk flags. Initially, it was iris flags. So it was possible to know the eye color of the person. In India, we don't have that much variation of the eye color, but if you go out of India, there is a wide variety of eye color available. You can see here in picture so many eye colors and pigmentations are available. So based on this 6 SNPs, which are affecting the melanin synthesis in the retina and in your this eye, basically you can have this particular the color combination, and you can identify the color of the eye based on DNA. So when you are having only skeleton with you or a part of bone with you, you can still predict the eye color, the skin tone, and the hair color of the person. So nowadays this particular high risk flex system is available for your skin tone, for your eye color, and for your high risk hair color. So H high risk means H for hair, Irish for your eye, and S for your skin. So skin tone, eye color, and hair color. Because each and everything, the coloration of these 3 things is basically responsible. It is because of the melanin synthesis in your body. And the metabolism of melanin synthesis is dependent on SNPs. So they have analyzed these SNPs, and these predictors are available. So if you can visit this website, Erasmus mc.nl, that is wonderful website. If you will visit this, you will get much more insight into this, because I cannot discuss everything here because it is a very wide topic. But yes, this is the modern tool which is still to be applicable, or you can say exploited in the Indian forensic science scenario. Then the microbial forensics. So PMI estimation, post-mortem diagnosis of drowning, soil identification, pathogen detection, and bioweapon identification. Here the microbial forensics works. Right, nowadays you can see the corona. Right, so virus was detected by qPCR. So the same qPCR which we are using for the DNA quantification can be utilized for the microbial analysis as well. You can see I have shown here the degradation stages of a body, or you can say the carcass. And these degradation stages can be known by bacteria also. Right now, people are using entomological aspects to identify these different stages, right? Blot stage, advanced degradation stage, and different stage. But after the skeletonization, it will be very difficult to tell that how much time has been passed, post-mortem. So it is very difficult. But the microbial analysis can give you the rough estimation, or you can say the 98% estimation of the PMI up to 180 days. So this is a very, very big time, right? Then soil identification. So different parts of the soil are having different variety of microbes, and these microbes vary from, definitely seasonal variations will be also there. But if you are having a database, properly managed database of seasonal variations of that particular soil, you can identify, because many a time, the smuggling, and you are having, the vehicles which are involved. So you can have the soil on the tire of that particular vehicles, and you can identify where the soil come from, or you can say even many microbes are unique to certain environment, and you can tell that okay, this is not the microbes which is the native of this place, and it can be, it can give you much lead about that. And finally, pathogen detection. So microbial forensics is having a tremendous application in pathogen detection, because nowadays most of the pathogen detection is based on the PCR or molecular biology. So even in forensic cases are also, it is very important to know about food poisoning, or the poisoning related or food industry related. You can use this biovapor identification, because of bio warfare, right? The many theories are also going on that corona is also bioterrorism kind of activity, but no one can prove. But this is the field microbial forensics can be utilized to identify these kind of things. Post, I told you that in WTC, they identified the strain, that the strain was stolen from the US laboratory only. One of the laboratory which was growing this Bacillus anthracis spores was means because each and every bacteria which is utilized as a bioweapon will have some signature sequences, and those signature sequences can be identified and can be utilized. Means this kind of analysis can be utilized for the forensic investigation. This barcode of life database, I think many of you, those who are working in wildlife forensics, are having this particular tool, handy tool, available on the website. And this is a wonderful database of the biodiversity of whole world. And whenever you are having any kind of insect or any kind of animal, if you want to identify, this is available, barcode life of database. Just you compare the cytochrome oxidase 1 sequence on this database, just like NCBI, blast of the NCBI, you can do the blast on this database bold, and you can identify your animal using this particular database. And the final modern technique that I will discuss, and then we'll directly go to the limitations, or you can say the challenges of the DNA analysis. This is very interesting part of the forensic analysis, molecular autopsy. I think autopsy is known to all of you, but this is a molecular autopsy. This is case one in February 2000. What happened? One child was dead because of, you can say, the fluoxetine related death. Basically, it is a supplement, fluoxetine. It's a drug which is given to the child for their particular growth and everything. But because of one damaged gene, cytochrome P4502D6, basically this gene is very much important for drug metabolism. Not only that drug, but many drugs are metabolized by many alleles of this particular gene. So this gene is highly active for the drug metabolism. So this is a lower metabolism. So when the child was not cured with this particular thing, it was killed. The child was dead. And later on, it was known that child was having the genetic deficiency of this particular gene, and that's why he was not able to cure. Right. Then this case, it's a very interesting case in 2006. In August 2006, it was published in Lancet. Right, pharmacogenetics of morphine poisoning in a breastfed neonate of a codeine prescribed mother. What happened? The mother was prescribed codeine drug because of the C-section, because of to relieve the pain of that particular C-section. So 13 days the baby died. So when the baby was means the postmortem was done, it was revealed that it was the morphine intoxication. So then they were surprised that what happened. So whatever the mother who preserved the milk on the 10th day, right? That milk was having the concentration of morphine more than 20 mg per liter, right? And that is very much high concentration of that particular morphine. And they suspected how this mother milk was having that much morphine concentration. When they investigated that, at that time, it was revealed that mother was having, the gene duplication of codeine metabolism, CYPD. Mother was having, hyper metabolism for CYPD. And that's why whatever the codeine was there was completely converted to the morphine, and the morphine concentration in milk was very high. And finally, that neonate was dead. So it means mother was the killer of her own child. So these kind of cases where the death cannot be explained, the COD and MOD is not, you cannot tell about the COD and MOD at that time. This kind of autopsy, this kind of molecular analysis can give you a really big sight. And nowadays you can see the cost of the total whole genome sequencing is also very low. It's very low nowadays. Regularly, people are advising that okay, you do your genome sequencing and that will prescribe you the drug, because it may depend on your. So that's why based on this paper, the whole you can say the gynecologist community was alarmed to prescribe this drug to the mother with C-section, because they have to check whether the mother is not hypermetabolic for this particular drug. Otherwise, it will not be good for the neonate, because it will turn into the you can say this kind of accident, right? So this field is need to be explored, and this field is very much a very good tool. See, this is not, you can see this is the case of 2020 year back. This is the case of 2006, right? It's 14 years back. So we are now in 2020, 2020, and what are the tools at that time? This analysis was costing hell lot of money, but now this kind of analysis is done very at a cheap rate. So why not to go for that, right? So this kind of analysis is very much. Then what are the challenges? So I'll just sum up these challenges in 10 minutes. So these are the factors affecting the biological samples. So these are, you can say, the bottlenecks, or you can say the hurdles to any biological system: temperature, humidity, bacterial contamination, and time. So you have to store the material at low temperature to arrest the microbial activity and to arrest the chemical reactions. Humidity should be avoided. Why? Because the water activity will enhance the bacterial growth. And I told you that none of the chromatin will be sterile. So that's why microbes will be always there, and they will grow if the humidity is there and temperature is there. So if you will avoid both these things, you can control the bacterial contamination. So while collecting the samples also, you have to be very careful that you minimum, or you can minimize the taking the bacteria in your sample. And that finally time, there is no limit that how much less time, as soon as possible. 5 minutes, perfectly fine. 4 minutes, better, right? So as soon as possible, right? You should reach the laboratory, your sample should reach the laboratory for the analysis. Determining the sources of DNA, that is also, because of the nowadays, the sensitivity of the PCR, even a single cell will give you the profile. So that's why when you are collecting the sample, for example, the case of this particular, I'm giving you this exhibit, right? So this weapon, basically this is used for daily, chopping of the vegetables. So this handle is utilized by the lady who is living in that house. Maybe she may be using, even the person, the male may be also utilizing for the chopping of the vegetables if he is helping to his wife, or even the children are also using this. So you are depositing your cells. It means the whole family has deposited the cells. And finally, this weapon was utilized by the third person to kill that person in that particular family. And when you are taking the touch DNA, or you can say the DNA from this particular weapon, right, and you are finding, 5-4 profiles. So at that time, whom to blame? Any family member or any third person, right? So this is called, the you can say challenge. The mixed profile challenge is there, and that's why you have to be very careful while collecting the samples. And sometimes if the mixed profiles are there, many softwares are available for the analysis of the mixed profile, but still the challenge of the mixed profile is there, and it is very difficult to analyze these mixed profiles. Interpreting the trace DNA evidences. It is one of the greatest challenges in forensic science is the presentation of the evidence in court when it is a trace DNA. Because in trace DNA, there are a lot of limitations which are there, which is not acceptable in the court. Or you can say sometimes courts or defense lawyers are very much learned. So at the time they will ask you the question about some scientific limitations of your technology. At that time, court will not believe your data, and your evidence will be out of court. It will not be accepted in the court. Neglected questions. Yes, these are the few questions. How was the sample deposited? Nobody is questioning how was the sample deposited. Nobody is questioning how long will it persist there. So, we collect the samples. We never know. We never ask that, okay, whether we should collect these samples or these samples. When you are having a choice, at that time you should be well known, or you can say you should be well knowledged or well informed that, okay, this material should not be collected because it is completely degraded, and it might not have proper DNA, but it might have proper DNA. Will it be transferred further? So, secondary transfer issues are also there. And how can it be detected, recovered, and contaminated? Even, for example, you are having some very challenging sample you have never seen in your complete tenure, such kind of, and how to collect the sample. So at that time, you should use your intellect and your knowledge to recover and collect that particular samples. Finally, last but not the least, the communication or knowledge gap between crime scene investigators and laboratory scientists, and it is huge in India. You all must agree with me that the investigation officers who are working on the crime scenes are having some different knowledge, and the persons who are working in the lab are having the different knowledge. And that's why the lack of knowledge to these persons will lead to the faulty collection of the samples, and finally will lead to the faulty analysis of the material, right? And this is a huge gap, and that's why the university like GFSU, right, is doing a lot of efforts in this particular area, that we are training hundreds and thousands of people, or you can say the persons, police officers, criminal justice systems, and many more persons who are involved in justice delivery systems. We are training them. Last year, more than 10,000 officers were trained by GFSU. So this kind of knowledge gap should be reduced, and these persons should be trained in a serious way to get the result in a serious way. If they are trained, then you are going to get the clean samples, and your conviction rate may become increased, or you may get the success ratio very high. It means out of 80%, you can reach up to 90-95%, because many police officers are not knowing the difference between biological samples and non-biological samples, and that's why sometimes they commit mistakes, because obviously they will commit. If I'm not knowing, then definitely how can I differentiate this kind of things? And proper implementation of the SOP, standard operating procedure, will not be there on the crime scene, and it will ruin the crime scene, and finally it will ruin the lead, and finally the justice may be delayed, or may not, maybe denied also many a times, and the person perpetrator may may walk free, right? So this is the situation nowadays. This is one of the most important limitations in India, and we have to fill this bridge and fill this gap between this. So that's all. These are the few references. I would like to go through this book, Forensic Biology, second edition by Richard Lee. John Butler, obviously, for forensic DNA typing, John Butler is always there. This is also a very interesting book, you can say, from Dragan Primberg, that is Forensic DNA Applications, which was published in 2014. And I think the next edition is also going to, I think in 2021, it will be there. The latest edition of the Butler is Interpretation, Advanced Topics in Forensic Typing Interpretation. I have not cited here, but that is also a very good book for those who are advanced learners, right? Otherwise, those who are at graduation level, you can start with these two books, and you will have a very good knowledge about the forensic DNA fingerprinting. So thank you very much for being a wonderful audience, and now you are free to ask the questions.
Thank you, sir. Thank you
Hello,
so much. Sir, we
Good
have,
afternoon,
Yes.
sir.
dear participants,
Sir,
you can
I
ask
have
the
a
question
doubt.
now.
Okay.
You were,
Okay.
you were telling about the sources of DNA and mentioned about the knife, right, sir?
Okay.
So, if those people wash that knife, so the evidence won't be there, or will it persist?
See, it depends on the washing method. For example, if you are washing only normally with the water, the chances are there that your DNA will be there. But if the knife is washed properly with the detergent and something, then there are very less chances that you will get the DNA from the knife handle.
Okay, sir. Thank you.
Good afternoon, sir.
Good afternoon.
Sir, my name is Inderjeet Singh from MIT Institute of Forensic Sciences.
Yeah.
I want to ask you a question like, what are your views on AFLP and mito-DNA analysis of, kind of applications of that?
AFLP is amplified fragment length polymorphism. So AFLP, I have rarely seen AFLP being utilized for forensic analysis. But yeah, of course, mitochondrial DNA analysis having a tremendous scope, and even you can know about the familial relationship using that mitochondrial DNA. Though in India, very few labs, or you can say the 1 or 2 labs, are utilizing this facility for mitochondrial DNA analysis. But yes, if when you are failed with the STR profiling, you can go for the mitochondrial DNA, and at least you can find the lineage, you can find the family. You cannot pinpoint the person, but you can find the family relationship.
Well, thank you so much, sir. And sir, what do you think about the Y chromosome analysis?
Y chromosome analysis is done in mostly the rape cases and paternity analysis. So when multiple persons are involved in rape case, at that time Y chromosome analysis is done to distinguish these 2% or 3%. So it will further help to segregate the mixed profile. So that's why it is used. And in alleged father, sometimes when the case of because the Y chromosome analysis cannot be applied when the children female, right? When the child is female, but when the male child is there, definitely the Y chromosome analysis can be utilized.
All right. Thank you so much, sir.
Hello, sir.
Hello.
Hello. Myself, Sarad Mittal. Sir,
Yeah.
normally the animal virus contain the DNA, but why this COVID contain the RNA is there?
See, I'm sorry to say, because I'm not a virologist, but it depends. Viruses are having it is coming from this HIV family, so it is a retrovirus. So all
Yes,
the
sir.
retroviruses are having RNA. So it is not true that every animal virus is having DNA only, right?
The
So
3 or
many
4
animal
virus, yes, sir. 3 or 4 animal virus contain the RNA normally.
yeah.
Maximum contain the DNA, sir.
Yeah, this is true, but corona, whatever this is corona, it is nothing but the upgraded version of the SARS only, right? So this is the having RNA. So most of these HIV is also having RNA. So this particular
sir.
virus is having RNA.
Yeah. Thank you, sir.
Yeah.
Dear participants, anyone else? Okay, sir, we have some question in chat box from Mr. Venkatesh. Actually, he has 4 questions. First one is, through airwax we can get DNA for criminal investigation.
MVAC.
Earwax.
Earwax. Yeah. Earwax is also a good source of DNA. We can get the DNA. There are cases where it is even earwax is also nowadays the phenotyping is also done, because earwax pattern is different, dry earwax and wet earwax. So many papers are also available for dryness of the earwax and the wetness of the earwax. So earwax is also a good source of DNA.
Sir, the follow-up question is, if we get DNA from earwax, either it can be used for investigation purpose, or is there any other technique for extraction of DNA from earwax?
No, earwax, see, whatever the earwax is there, it is simply extracted by whatever the established technologies are there. Just you have to process the sample properly. Otherwise, earwax doesn't contain that much PCR inhibitors. So that's why it is considered as a body fluid only, right? And very easily DNA can be extracted from the earwax.
Okay, thank you, sir. Sir, the next question is from Diljan Khan. The question is, which DNA plays a major role in child identification, paternal DNA or maternal DNA?
So is he asking about the sex of the child, or he is asking about the identification of
The question is
father?
like that, the general question, the child identification.
So in child identification, see, the mother and father's DNA should be required, right? And when you are comparing, the 50% alleles will be from the mother and 50% alleles from the father. If a single allele is also not matching, you cannot tell it is a real biological father. So the father's DNA and mother's DNA both is required. But yes, if the child is male, then you can directly use the father's Y chromosome and children's Y chromosome, and you can compare directly with the Y chromosome also. So it depends. If it is a female, then you have to use autosomal genotyping, and if it is a male child, then you can use Y chromosome also for the father's part. And if you want to identify the mother, then you can use mitochondrial analysis. Right, in that mitochondrial analysis, directly you can link mother with the child.
Thank you, sir. The next question is from
Hello.
Miss Anina. MVAC
Mm-hmm.
can be used on rough surfaces like a stone
Mm-hmm.
if it can also
Mm-hmm.
used in collection of DNA from old aged crime scene.
Yeah, because whatever the vacuum creator is there, right? So if it is, because it is a very good vacuum system, so the rough surface is like cloth. But if it is having a very uneven surface, then it will not be applicable. So at that time, you have to wash the stone or that surface, and then you have to collect. That is the only option. But if the surface is not that much uneven, if it is roughly flat, then you can use this particular system.
Thank you, sir. The next question is
Uh,
from
so,
Mr. Saurav Karn. What is the
Mhm.
difference in method and protocol of extracting RNA from whole
Mhm.
blood versus serum?
Okay, so I don't think so. There is a difference to get the RNA from serum or blood, right? Uh, so see, right now, for example, in COVID analysis, we have also established COVID-19 testing in our lab. So whatever the kits we are using is can be utilized for serum also and can be utilized for blood also. Ultimately, you have to lyse the blood, you have to lyse the serum, right? That's all. And it is basically, see, what any DNA, or you can say the DNA extraction system, cannot differentiate between DNA and RNA. It is actually extracting nucleic acids, right? If you are interested in RNA, you are treating the solution with DNAs, and if you are interested in DNA, you are treating it with RNAs. So this is the post step or pre step during lysis also and after post lysis also. Ultimately, any extraction system is extracting nucleic acids in general. It cannot differentiate between DNA and RNA, right? And finally, you can get nucleic acids palated, and if you are interested in DNA, you can treat it with the RNAs. If you are interested in RNA, you can treat it with DNAs. So DNAs will be degraded, and you will get the RNA. So this is how it is basically happen. So most of the time, when it is meant for PCR, people generally don't bother about the degradation of RNA or DNA. They extract the complete nucleic system, nucleic acids, and they apply it for reverse transcriptase or normal PCR in case of RNA or DNA respectively. So it depends like that.
Okay, sir. Another follow up question is that for crimes in genetics, apart from amyl Y and amyl X, are there any other
Mhm.
genetic marker used for sex determination of an unknown?
Yeah, see, out of amelogenin, there is a Y chromosome. Y markers are also available, which can give you the sex if it is male is present. But yeah, you cannot, apart from amelogenin, none of the marker can distinguish between male and female both.
Thank you, sir. The next question is from Chirag Thakur. What are the methods
Yeah.
of detection of Y chromosome in mixed profile?
Uh, it means when we are having a Y chromosome and at the same time you are having the mixed profile,
Yes, sir.
right?
Yes, sir.
So at that time, see, recent analysis is telling that up to 3% you can identify. If it is more than 3%, it will be very difficult to identify. So softwares are there, algorithms are there, but when the markers are more, so it will be very difficult to tell which marker comes from which person, right? So that's why the latest whatever the article I have read till now, so 3%, maximum 3% can be identified, not more than that.
Okay, sir. Thank you, sir. The next question is from Miss Varsa. Can we collect any DNA or RNA from completely charred tissues?
It is very, you can say, subjective question, because if it is a chart, so you cannot tell that you will not get the DNA, right? So you have to take the chance, because many a time, if it is completely burnt, but sometimes what happens, because of the tissue and because of the bone structure, some internal part remain intact that we don't know from outside. If it is completely charcoalized, it means completely charred, and you are having completely carbonized matter, then it is not possible to get the DNA. But yeah, if you are having some hard structure which is available there, then I would suggest that definitely you should go for at least one chance to give the DNA extraction by whatever the modified method or whatever the suitable method you are having. It may be possible that if you are lucky, then you may get the DNA, right? So, but when it is completely carbonized, then there is no mean to get go for the DNA.
Thank you, sir. We will take one last question.
Okay.
It is from Mr. Sariful Alam. In a gang
Mm-hmm.
rape, is it
Mm-hmm.
possible to DNA identification all person related to this crime?
No, I told you no, that up to 3 persons we can identify. If more than 3 persons are involved, it is not possible. You have to take help of corroborative evidences, eyewitnesses, or some other evidences which can lead to the more persons identification. For example, more mobile tracing, or mobile tower tracing and some data tracing or like that. By DNA, you cannot identify more than 3 persons when it is a mixed profile.
Okay, thank you, sir. Sir, actually, we got one more question from Dr. Manu Smriti.
Mm-hmm.
Can you put some more light on retrieval of DNA from paraffin section?
Yeah. Actually, recently, not recently, but I think before 1 year, we got one very interesting case. 18-year-old paraffin block was provided to us to get the DNA from that particular paraffin box. So when we used the normal, actually there were 7 blocks with us, because the person died with cancer and multiple biopsy was done, and that's why we were having so much blocks with us. So we utilized one block, and we tried to isolate the DNA, but we could not. It means we used first kit, that is a Qiagen kit. Then we used almost 7 different kits and tried to get the DNA, but it could not be done. Then what we did is we take the different tissues which were entrapped into the that paraffin. We washed it till multiple times with multiple solutions, and we did completely R&D. You can say a completely array of experimental design we did, and finally one combination worked. Right, because I cannot reveal right now because it is under publication. But one combination worked and it revealed the DNA. I cannot tell that okay, because of that combination DNA was isolated. It may be possible that that part of tissue was not that much damaged, and that's why we got the DNA. And we got the DNA from 18-year-old paraffin embedded block. So the how you are processing your tissue, it is very much important in paraffin block. So that's why you should not directly blindly follow the protocol, because sometimes some user developed protocols are also available on the web, right? When you are searching this Google, or you can search in even for example, if you go on Qiagen website, many user developed protocols are also available. So you should try all those protocols also when you are having some challenge samples. Right. Even moreover, if you will, put your question on ResearchGate or some scientific community, definitely some people will help you out with some, modification in the protocol, and you will get the, uh, you can say, yeah, the success of that particular challenge samples. Right.
Sir, the follow up question is the scenario of DNA database, is it maintained in India?
No, we are not having DNA bill yet, which is passed from the Rajya Sabha. It is the process is going on. Very soon we will have DNA database, but till now we don't have any DNA database.
Okay, thank you, sir. Thank you so much. Dear participants, if anyone have more question, you can ask right now.
Hello.
Hello.
Good afternoon, sir. So I do have a question regarding the identification of person or the murderer who recently had the plastic surgery and the hair transplant on him. So how do you identify that person from the crime scene? Uh, like from the DNA or from something like that?
Plastic surgery means you are telling about the skin is grafted from another person.
Yeah, sir.
Yeah, so this is called a case of chimera, right? It happens many times. Happens. It means, are you sure that the skin was transplanted from another person?
So I'm just asking us. So it is just out of curiosity, what will happen in
Yeah,
such
yeah, yeah.
cases?
You are right. You are right. But in such cases, mostly the skin grafting is done on oneself only. It means skin is grafted from other part of the body to that person because the skin rejection will be there. But if it is possible, see, theoretically we believe, right, as an instance, then it will be difficult to identify because this is a very important, it's a very interesting question because the chimera exists, right? The persons who are getting blood transfusions regularly, like thalassemia persons, or the persons who are suffering from some any blood related diseases, and they are undergoing frequently under blood transfusions, so they also develop slowly and slowly some major profile in their blood, right? So such kind of persons, the skin you are talking about the plastic surgery, right? And that part is available on the crime scenes, so it will be difficult to find out because you will get two profile, because that person's profile and that person's who got the grafts from other. So it will be difficult to tell. Yeah, but sometimes what happens, for example, you are getting a blood transfusion or such kind of grafting. So in DNA profiling, you will get a minor profile and you will get a major profile, right? So pics will be more of yourself and the pics will be smaller of other persons from where you got the blood transfusion, because the amount of DNA or amount of the cells from that transfusion will be always low than the your original blood. So this kind of analysis will require some more rigorous analysis of the DNA profile, and then you can come up with the solutions. But sometimes, when you are having a bone marrow transplantation or some such kind of transplantation, at that time, after over a period of time, you will have an equal profile of the persons. So at that time, this is called a challenging chimera. So this kind of situation may occur, and that will be very difficult to analyze on forensic scenario.
Yeah, sir. So I do have second question regarding
Yeah,
my
yeah.
curiosity.
Mm-hmm.
So the other question is regarding so using knife, the ice knife, okay, the knife made up of ice, and
Ice, yeah.
if someone use it with their bare hands, and he's the murderer and seen like that, so how he gets away from those scenarios, or he could be convicted, or he could be like found in those traces of the waters and things like that.
Yeah, it will
Is
be
it possible?
very, yeah, it is possible, but you will have to dig in a detail, right? So whatever the water is there, if you are you will use amvac system and you collect that water, and then because it will collect the other cells also which are available on the for example, others who even enter the room, because it is very sensitive. PCR is very sensitive, so it will be very difficult if such kind of modus operandi is there for the murder and such things. So it will be very difficult to get the DNA from that. Yeah, but other things, for example, if the person has scuffled with that particular person and some other part of the body is bruises with this part of the body, and then you are getting the things, it may be possible. But with that particular kind of scenario, it will be very difficult.
Okay, sir. So thanks a lot for the session and for responding the answers.
Yeah, thank you very much.
Okay, dear participants, anyone else have question? Sir, so we will conclude this session.
Thank you
Thank you
very
so
much.
much, sir,
So
for.
yeah,
Yes, sir. Can you answer?
so yeah, it was my pleasure to be here, and thank you to the Centurion University who collaborated with the GFSU for such kind of